Hello, welcome back to the Computational Genomics Lab. Today we are going to do Level 9, Lesson 2, overlapping peaks. So we made a function last time to import peaks, and this time we are going to make a function to find overlaps between the peak files. So at the end of this, our goal will be to take the 12 files we have from different time points of dox exposure and find peaks that are in common in all of those 12 files. So that could be pretty handy. That means those are really robust sites of open chromatin that are always there in a stem cell, and you could think of some interesting questions to ask, whether if a gene has a peak open, is that gene more highly expressed in a stem cell, which you have that RNA-seq data in the TPM plot. So you could ask, are they higher average TPMs if there's an ATAC peak or not? And that's where we're going to be going downstream. But let's get started with doing overlap. So we're going to go to master class level 9. We're on O2, and I am going to load this in my RStudio here. O2 peak overlaps across samples. And go to the top. I've cleaned my environment here. So we need to source our functions, which is the find common peaks and find my peaks. We're going to make find common peaks right now. And this is just a goal to learn the lesson of how to do overlaps. And really you just need at least 2 files. And we did that last class, that we can just overlap them and find peaks that are in common. And here we can do take one file, do an overlap, take the next file, do an overlap, take the next file, do an overlap. And each time repeating it with, after the first overlap, only taking the peaks that are in both those files, overlapping that with a third file and a fourth file and a fifth file. So you'll get down to just the peaks that are there. That's what we're going to do now. So the peak path is where your files are, your .broadpeak peak files. In the description below, there's a place for where those are freely available. You can download them on a folder and get the file path to that folder, and you'll be able to follow along from here. Okay, so there's where my peak files are. We're going to do make this FL object, which is just all the 12 files we're going to upload of all of our peak files, and we're going to put that into import peaks, the function we just made last class. And now this is going to take about 1 minute to upload, and I have in here, this is redundant from the last class as well, that this is just going to tell you the number of peaks in each file if you wanted to keep track of that. It's about 60 to 70,000. We already saw that, so we'll skip over that. And we did this as well, where we can take I'm just repeating some of these steps that we would do, is we would make a file path to a specific folder, match only to files we want that are end with .broadpeak, and then this sapply will go through and only extract those, make a sample name object of the wild type and control and the time point. Okay, so find common peaks, that's just a review of the function we did last time. We're going to kind of switch gears to do a function that takes each peak file, the first peak file, finds and overlaps, takes that overlap, maps it with the third file, takes that overlap, maps it with the fourth file, until at the end you only have files that overlap in all samples. Let's do that. Okay, so we're going to make this function called find common peaks. So you can give it, we right now have a list of 12 in my peaks here, but you could give it like just 2, or we could grab out using a grep just a couple of these files. So a good strategy is to always upload as many files and then pick from that as your main source of your analyses. So here we're just double checking to make sure the input into this function is a list of GRanges, as my peaks is currently a list of 12 GRanges. So it's just going to make sure if it's not a list of GRanges, then stop and say input must be a list of GRanges. So it's just a nice check if not all the things in the object that's inputted are GRanges. Okay, so to find the common peaks, we're going to make an object that's currently empty, and we're going to start with the first peak file in the list. So this will simply be KO control 0, doesn't matter. It's sort of arbitrary. It's not totally arbitrary, but for now it's okay. We're just working on the concept of multiple overlapping files. So we're going to take the first one, and then for the second through the rest of that list of 12, we're going to make an overlap. And so we're going to make a GR list of I, which is the second file now, because we're doing 2 through the length of the file of this mypeaks 12 files here. So we're going to start with the second one because we already have the first one indexed here. Okay, and then we'll do find overlaps, which is the genomic ranges function, to find that in common peaks and current GR. So it's going to take the current common peaks here is going to be the overlap between the next file, and then those overlaps are it's going to be a reduced number of peaks, and then to overlap again you're going to be limited to that number of peaks, and you're going to go less and less and less each overlap that occurs. So that's what the current GR is the second, the third, the fourth, the fifth. But the new one is the common overlaps object. Okay. And then we can use subset by overlaps to just reduce it to common overlapping regions and kind of not worry about the @to, @from we went over. You can dig into it more, but basically it will give us the a more easily accessible set of overlapping peak intervals of a chromosome start and stop, and where the two files had an intersecting peak. Like if they overlap perfectly or like that, they would be counted as one region of a peak. And that's what subset overlaps is going to do. And then for each of the peaks currently have a name in them, but now they're going to be different because each file has a name of a peak, but now that we've merged them, there's really no name for them. So we're going to rename them at the end called common peak underscore, and then the name of the peak as it is in common peaks. So for each file, the end of the overlaps, our peak names are going to be have a chromosome start and stop of where a peak occurred in every sample of the 12, and we're going to name each of those peaks common peak 1 through however many peaks there are in common peaks. Okay, and then return common peaks. So let's run that function, set it up. So now that's loaded, or we could source the function file, which we did. And so we can do find common peaks for mypeaks, and this is going to tell us the overlaps of peaks that are in every single one of our samples. These warnings are those GL chromosome things, and if we look at this, we can see each peak now has a number called common peak, and that means they're common in all the samples at 0 all the way through the time course of this analysis. Okay, so now you could clear your environment. I'm not going to do it because it takes a minute to load those things. Set the peak path, load this function, make an FL. That needs to be done, and then use that to use your import peaks function, which we did last time. Imports a set of GRanges that can now be used in find common peaks. So you could have just loaded the two 0 time points and used common peaks, and would have found the same thing we did last time by taking the two files and using find overlaps between them. So I think you can now take those out and take a look at them in IGV and see what you think of those peaks, if they look good. And at the end of 09, at the end of this level, I have an RMD file that shows you those peaks and the figures of them. But now you have a handy function that you can put in any 2 GRanges or 100 GRanges or 1,000 GRanges, and it'll find the overlapping regions that occur in all of those peak files. And so next, what we want to do is say, okay, cool, now we know where all the peaks are of open chromatin. How do we map those to protein coding genes? And we'll use GRanges and find overlaps with genome annotation features and put it all together in the next class. All right, see you there.